without further ado, I think we can invite our second speaker for this session, Associate Prof. Dr. Noor Baity Saidi from University Putra Malaysia, with the title Soil Bacterial Communities and Physicochemical Properties Associated with Fusarium Wilt Disease of Banana in Malaysia. I think it is sort of related to the previous presentation. So Dr. Baity, the floor is yours.
Okay, let me share my screen. Okay, is it on now?
Yeah, we can see your slide.
All right. Thank you very much to the organizer for the opportunity to share a little bit about banana research from our lab. We have listened and learned a great deal about microbiome just now from Dr. Paul. So in the next 15 minutes or so, I'll be sharing about the soil microbial communities of banana in Selangor, Malaysia, and its relationship with the soil physicochemical properties. I have to mention that this is our first experience dealing with metagenomics data, and we are still learning. I guess with metagenome data, we can only make predictions, and much more data is needed to strengthen the links between the sequence and all the functional predictions that we have here. So nevertheless, let's look at where we are now with banana production in Malaysia. So Malaysia is doing quite well. Actually, in terms of SSR, we are at 98.7%. While we are self-sufficient in our banana production, we still import some bananas, primarily from the neighboring countries, the Philippines, and also Thailand. Malaysia produce more than 50 local varieties, local banana cultivars, and among them, pisang berangan and Cavendish are the main cultivars that are planted in the country. They made up about 50% of the total production of banana in Malaysia. Unfortunately, they are also susceptible to the Fusarium wilt disease. Earlier Professor Aikin has also mentioned about the disease in her talk. So we know that Fusarium wilt is caused by soil-borne fungal pathogen, Fusarium oxysporum, specialis cubense FOC, and there are 4 races of FOC, and the most virulent one is TR4, at least in our region. And so we can see from the images that FOC attacks the vascular roots of the banana plants, and it blocks the vascular system. And TR4 was first discovered in 1990s in Malaysia and Indonesia, and from April 2021, TR4 is currently confirmed in 23 countries. The invasion of the vascular tissue through the roots causes discoloration and wilting that first appear in the older leaves, eventually killing the plant, while internally the vascular discoloration will become brown to almost black in later stages of infection, and it can be seen in the pseudostem and also the rhizome. However, there are no symptoms present in the fruits. Right, so diseases caused by soil-borne pathogens are notoriously difficult to control for several reasons. Many soil-borne pathogens produce persistent resting structures, such as the chlamydospores, that can survive in the soil for many years, even in the absence of susceptible hosts. Control measures using chemical fumigation is unsuitable for large-scale application. Application of pesticides or fungicide is often insufficient because of the poor accessibility in soil metrics. And to make the issues worse is that there is no effective fungicide or other eradication method presently available for Fusarium wilt. So I'm just going to skip this slide since Dr. Paul has already explained on the microbiome and microbiota. Right. So the idea of tackling soil-borne disease with a microbiome can be based on the facts that number one, microbe interacts with one another, and number two, plants employ a palette of mechanisms to modulate their microbiome. And the grand concentration of the microbial life, which as Paul explained just now, is a mixture of beneficial and also pathogenic microbes, and they lies in the thin soil layers surrounding the roots, known as the rhizosphere. Exudates from plants. So the microbial composition in the rhizosphere could be different from bud soil. And pathogens, they are negatively affected by the co-inhabiting microbes through several mechanisms such as antibiosis, and also they compete for nutrients. In many reported cases of Fusarium infection, symptomatic and asymptomatic bananas have been found in the same crop fields, suggesting a unique interaction may occur between the plant and its niche environment. And we hypothesize that different bacterial community structures and specific soil properties are associated with banana health status. So the objectives of this study were to examine the rhizosphere bacterial assembly of symptomatic and asymptomatic bananas compared to bud soil in a banana farm in Malaysia via 16S rRNA high throughput sequencing, and to compare soil physicochemical properties between healthy and also Fusarium infected soils. As for the methodology that we applied, the soil samples were collected from a 5-acre banana farm in Sabak Bernam, Selangor, cultivated with susceptible variety, CV Berangan, for more than 2 years. We have obtained permission to collect the plant and soil samples from Sabak Bernam District Agriculture Office. So these are images that we captured during the sampling, showing banana with Fusarium with symptoms and also the non-symptomatic plants from the same farm. Right, so we collected plant and soil samples. Plant samples were used for the detection of TR4 via PCR, and soil samples were sent for sequencing and also soil analysis. Yeah, we collected rhizosphere and also bud soil for the analysis. The sampling was conducted in a completely randomized design, and the plants were at least 10 to 15 meters away from one another. There are 5 replicates for each group of soil samples. There are 4 groups of soil samples. The healthy rhizosphere, the healthy bud soil, infected bud soil, and also infected rhizosphere. So in total, we have 20 samples together. So let's look at what we have from these sequencing results. All right, so this figure shows the refraction curves of individual soil samples from the banana farm that we study. So the curves were near saturation, indicating sufficient sequencing depth to cover the bacterial diversity within individual soil samples. So interestingly, the infected soils, both rhizosphere and bud soil showed a higher number of OTUs than the healthy soils, the rhizosphere healthy and also bud soil healthy soil. So bacterial sequences of healthy and infected rhizosphere and bud soils were assigned at phyla level, with Proteobacteria, Actinobacteria, Acidobacteria, and Chlorophyllaceae as the major phyla associated with all soil samples. And Proteobacteria was the dominant phyla in all soil samples, representing 26 to 48% of the bacterial DNA sequences. And across the treatments, Proteobacteria was significantly higher in the infected rhizosphere compared to the healthy rhizosphere. And similarly, in bud soil, the phylum is higher in bud soil at 31% compared to healthy bud soil. While in the healthy rhizosphere, acidobacteria was most dominant and significantly higher in the rhizosphere soil. Okay, so the phyla is not significant in the bud soil. Right, so bacterial communities were also evaluated using richness and diversity indices. So as a measure of alpha diversities, the richness and diversity were consistently higher in infected soils than the healthy soil. All right, so we can see significant differences in all the indices between rhizosphere healthy and also infected. Okay. It is not that common actually to observe high number of OTU and alpha diversities in infected soil. However, a higher number of bacterial OTU and alpha diversity indices in Fusarium with infected soil have been reported before. So we think that the higher abundance of bacterial OTU in the infected soil is due to ability of plants to alter their rhizospheric microcommunity in response to pathogen infection, and also may be influenced by other confounding factors such as the above ground vegetation and also the fertilization regime. And we also measure the changes in species diversity in all soil samples, also known as beta diversity, and we found that the bulk and rhizosphere samples displayed significant separation. Okay, the pink and blue points, regardless of the health status, and we look at the soil samples in the rhizosphere, and then we found that the rhizosphere, the soil samples from healthy rhizosphere was clearly distinguished from the infected rhizosphere as shown here. All right, so considering the significant impact of Fusarium wilt on the community structure in the banana rhizosphere, specific bacterial species or genera are expected to be enriched or depleted, and based on the LFC conducted at the feature level, and Acidobacteria and Xanthomodaceae is the most dominant genera in the healthy rhizosphere and also infected rhizosphere soil. And the analysis also revealed the presence of the genera Burkholderia, Burkholderia, Streptomyces, Baity, Rhizobium among the significantly enriched genera in the healthy rhizosphere soil. The presence of this species has been shown to promote disease suppressiveness even in the presence of pathogens, especially from the Streptomyces species. So now that we have seen the marked difference in the community structure of bacteria in the banana rhizosphere, we went on to check the difference in the predicted gene function of the soil rhizosphere community. So we found that all the KEGG functions were relatively similar in abundance between RH and RI, the healthy and infected rhizosphere, except for the ribonuclease E, which is involved in RNA degradation or metabolism. And so, the rhizosphere, the healthy rhizosphere contains more microbiome involved in RNA metabolism and also transporter pathway, and these are essential functions generally required for microbes. And we thought that their abundance in the healthy rhizosphere contributes to the general well-being of the bacterial community to fight off invaders. So the differences in the bacterial community composition can also be related to changes in the soil variables. In this study, only magnesium and CEC were significantly higher in the healthy soil. So magnesium is an essential mineral element for plants and microbes, and has been associated with Fusarium wilt disease suppression, while CEC is a measure of soil's ability to hold and exchange cation, including magnesium, and soil with high CEC was reported to be conducive for beneficial microbes such as bacillus to suppress Fusarium wilt pathogens. So in summary, the bacterial community composition and diversity differ between healthy and infected soil, especially in the rhizosphere zone. And notably, these three species were among the highly abundant genera in the healthy rhizosphere. And in the soils examined, the health status of the soil is associated with the level of magnesium and also CEC. So as for the recommendation, I think since Paul also mentioned just now that findings from one field may not necessarily represent the findings from other fields. So I guess it is I'm interested to analyze soil samples from various wilt hotspots and maybe suppressive soil, which I don't know if we have any in Malaysia, and then maybe we can consider direct approach as proposed by Burk et al. By using microbiome transplant or transplant microbes with beneficial properties or metabolites from selected microbes, or using an indirect approach by changing the environmental conditions in a way that microbiomes also shift their structure and function. All right, so I would like to thank UPM for providing the research grant for me to conduct this project. And also all the hard works were done by my master student, Mrs. Fatih Nadia binti Jamil, and also assisted by my collaborators, Dr. Amalia and Dr. Temizi. Thank you. I welcome comments and suggestions. Okay, thank you.
Okay, thank you so much to Dr. Noor Baity. There was a bit misunderstanding just now. We will have Q&A session for every speaker, 5 minutes Q&A after the talk, okay? So for Dr. Paul, I can see that you have answered some of the questions through the chat box, but if there's any more questions anyone would like to ask directly to Dr. Paul, we can have it now together with Dr. Baity. So I welcome participants to raise your hand, and you can turn on microphone and ask questions directly, or you can just use the chat box. There's about 4 minutes left for Q&A. So I'm kind of interested with these two presentations. It is sort of related to my field. So yeah, I can see that both of you suggest like microbiome transplant, right? So how do we go about that? Maybe Dr. Paul or Dr. Baity can give some comments on that.
Yeah, that's actually an excellent question. I mean, we've got to the point where as I said, we've got that list of most wanted core taxa, and at the moment we've been able to isolate about 2/3 of them. And so now we want to start introducing them to plants and get meaningful screening results. We've found that if you take tissue culture plants and you soak them, even in core taxa, which should be associated with plant fitness, very often the plants will die. If you plant them, then 99% of the time, the microbes that you introduce to plants will disappear once they get into soil. So it's actually a really, really difficult question. So yeah, we have discussions ongoing at the moment as to what might be the best way to do that. And effectively, we don't have answers. So we're designing experiments to find out what is the best way to get them in. At the moment, we have options at the tissue culture stage, and then we've got options at the hardening stage, and then perhaps once in the field. So yeah, but it's also interesting when you consider that when you compare So if you look at, say, Liz Aitken's group's work, and you consider like in resistant varieties, then FOC doesn't make it above the corm or the rhizome. So that would suggest that if you're going to introduce microbes into the pseudostem, you need to be sure that they're getting down to the roots. There's some kind of, there's the whole spatial and temporal ecology of the issue needs to be properly explored. So, sorry, just adding to the complexity of the issue rather than giving any solutions, but yeah.
Dr. Baity, any comments? We have a question from Prof. Yasmin actually. Or Dr. Baity, want to say something first?
No, it's okay. You can take questions from Prof. Yasmin first, maybe.
Yeah, okay. Prof?
I want to add to the confusion actually, because as a microbiologist, we know that microbes are very sensitive when you try and reintroduce. Do you consider that there may be some layering effects that you have to put certain microbes to sort of colonize first, and then you have a second layer of microbes, the layer of microbes, because that happens sometimes. Is that a possibility? One of the reasons why you cannot get, you know, total. It's a bit like the human gut, right? You know, the core 1, core 2, core 3. Do you think that might be the reason why it's so difficult to establish the an inoculated microbiome?
Yeah, I mean, we're certainly working on the assumption that a core assemblage is probably more protective than an individual core microbe. So a lot of the biocontrol strains that you see commercially available are often very ineffective. They disappear for whatever reasons. So I think, yeah, ultimately whatever it is, it needs to be dissected properly and approached with good science. So yeah, a lot of these questions like, are we likely to have more persistent colonization and better outcomes for growers if we introduce an assemblage of microbes at a particular life stage and then repeat doses at a particular frequency. So all of those things need to be, you know, properly studied and addressed to have a successful and reproducible management approach. Yeah, what do you think, Noor?
Yes, I guess I have not much experience with the application of microbiome of microbes, because this is like I said, this is my first exploration of microbiome of what we have in Malaysia. But yeah, I'm aware about the biological control, and then when we try to treat with microbes, so the bacteria that show promising activity in the lab, they must be at least fitting their characteristic when applied in the field. So the success really depends on our understanding of the how they interact with one another, and also with the host. It's promising, but it's very complicated, I guess.
Yeah, okay. I think we're a bit running out of time for this Q&A. Okay, very interesting topic. I think like we discussed a bit already, so why not we can have a further discussion together. Dr. Baity, Dr. Paul, Prof. Yasmin, myself, of course. And I saw just now that the Theo raised his hand, but I'm so sorry we can't take another question. We need to move on to the next speaker. I thank Paul and Dr. Baity. Okay, so.